In this video, we're going to go over the basics of some biochemistry, and specifically how to draw an amino acid or a protein, which is what amino acids get turned into, at a specific pH. And so you should have in front of you your laminated biochemistry sheet. It has a lot of useful information. I would hold on to this, especially if you go to college and you'll have to take biochemistry. So there are 20 different amino acids that make up proteins in cells, and these provide enormous combinations of different proteins that we could make. And so titin, which is a famous protein that is in muscles, there are 34,350 amino acids, and so that allows for 5.2 times 10 to the 90th possibilities. And the thing that's crazy is, see, that's more stars probably than in the universe. And so what does pKa mean? So let's look. Let's say we have a pK, and I'm just going to say 5.2. If you're above the pKa, so this is 50% protonated, meaning 50% of the H's are attached to your acidic location, and then 50% are deprotonated, meaning the H is gone. Well, if I'm at a higher pH, that's going to be more than 50% will be deprotonated. And then here, less than 50% being deprotonated. If you take an AP chemistry, one thing that you might make a connection to is that at the half equivalence point of a titration, pH equals pKa, and that's specifically this reason. 50% of your H's have been removed, 50% of your H's are still intact. Okay, so biological pH is 7.4, and what we want to know is our backbone. So group number 1 here is our carboxyl group. Group number 2 here is our amino group. Group number 3 is our hydrogen, and oftentimes we won't draw that in. And then group number 4 is what makes all the amino acids different. So groups 1, 2, 3 are all the same for every amino acid. And then group 4 is where the variation comes into play. Now we're going to get to it next unit, but you may see this wedge or these wedges and then this dash. I'm not worried about that right now. That shows us the 3D structure, which we'll get to in the next unit. So a carboxylate group at a pH of 7.4, which is how I've drawn this, is deprotonated, meaning there's no H there. Okay, that makes sense because if you remember from your pKa table, a carboxylic acid has a pH or a pKa of 5. Okay, the amino group is protonated, and that makes sense again. If we're at a pH of 7.4, well, when we're at above 7.4, more of those H's will be taken off, and specifically for an amine group, the pKa is 9 to 11-ish. Okay, this one's specifically 11-ish since the amine group is attached to an R group. Okay, and the overall charge of the backbone is 0. So if I look at it, it is 0, and that's called Zwitter ionic, meaning even though it doesn't have a charge overall, there is still charge within. This is going to be called our central carbon, which is going to be called our alpha carbon. Okay, and then this carbon, I don't have it here, but this carbon in the carboxyl group would be called a beta carbon. Okay, so you'll have to memorize these if you take biochemistry, and believe it or not, they're not very difficult. Now, what I want you to recognize is they all pretty much have the same backbone. The only thing that is different is the highlighted groups. And so that makes it a lot easier to recognize. Each amino acid has a 1-letter abbreviation and then also a 3-letter abbreviation, okay? So we could abbreviate glycine as G or GLY. Okay, and that'll be given on the test. Your amino acid pKa values, those will also be given on a test. So what I want to do is calculate the isoelectronic point, and that's specifically the pH where the amino acid is neutral. And if you have done gel electrophoresis, it would not move in an electric field. Okay? And so what we would do is we would draw glycine, and so glycine looks like this. And the R group of glycine is just a hydrogen. So what I would do next is I would look at my pKa table and I would find my glycine. The carboxyl group is 2.4, the amine group is 9.8. If I have 2 charges on it, what I would want to do is figure out the pH at which it is neutral, so I just take the average of it, and that makes it nice. Alanine, very similar, and I'm probably going to draw amino acids like this going forward. The R group here, I'm not going to draw the H because the H is always there, but the R group is just a methyl group. Okay, so this is alanine. I got to figure out the pKa's of alanine, 2.4 and 9.9. And so I take 9.9 plus 2.4 divided by 2. I get 6.15. Okay, so try out phenylalanine, try out methionine, and I'll put the answers here. Okay, now, when it gets a little bit more complicated is when you have side chain amino acids, which they all have side chains, but specifically side chains that have pKa's, like these guys, or any one of these. Okay, so this becomes a little bit more complicated. I'm going to draw cysteine. And the R group of cysteine is a carbon, then a thiol. And then I'm going to figure out the pKa's according to the pKa table, which I have in front of me. We've got 10.7, 1.9, 8.4, and so in a perfect world, it would be nice if we could take the pH or the pKa's and just average all 3 of them, but that's not what really works. And so what we'll do is we'll say that we have pH ranges. Okay? So here's what I'm going to do is I'm going to say, okay, 1.9, 8.4, 10.7. I want to find a pH less than all of them, so I'm just going to pick 1. Then I want to find a pH a little bit higher than 1.4, so I'm going to, but still less than the others, and I'm going to say 3. And then I want to find a pH higher than the next one, but lower than the last one, and so maybe I choose 9. And then I want to find a pH higher than all of them, and so maybe that would be like 11. Okay, so if this has a pH of 1, here's what this would look like. This would actually be OH. Why? Because we're lower than the pKa, meaning that this structure is best represented as having, or that alcohol group is best represented being protonated. Okay, now add up the charge. If we add up the charge here, we have a positive 1, neutral, and neutral. So overall, we would be positive 1. Okay, now I'm going to do pH of 3. How do things change at a pH of 3? This guy becomes negative. This is still the same, and this is still the same. And so if I add up all the charges, I have plus 1, minus 1. Overall, those are neutral. Okay, now let's do a pH of 9. Let's see how this is different. My carboxyl group is still the same. But now my thiol group, because it has a pKa of 8.4, it goes to being deprotonated. My amine group stays the same, and so when I check all of these, I add up plus 1, minus 1 is 0, minus 1 is negative 1. And then now I need to go to a pH of 11. That changes my amine from NH3 to NH2, and when it's NH2, we'll have no charge. And so overall that has a minus 2 charge. Well, what we do is we just take the average of the ranges. So what do I mean by that? Even though I picked 1, this is really going to be the average of the 8.4 and the 1.9. So I'm going to take 8.4 plus 1.9 divided by 2. And the isoelectronic point here is going to be 5.15. Okay? When you have a side chain, it is definitely more challenging to get the isoelectronic point. So let's try tyrosine. Tyrosine looks like this. And my pKa's is 2.2 equals 10.5, and this pKa equals 9.2. So when I get the isoelectronic point, before I can do that, I'm going to pick 2 of these pKa's and take the average of them, but I've got to find the range at which this would be 0. So my pH ranges. Okay, what's the lowest one here? 2.2, so I'm just going to say 1. Higher than 2.2 but lower than 9.2. Heck, you could say 7. Higher than 9.2 but lower than 10.5, I'll just say 10. And then higher than 10.5, that would be 11. And so what we need to do is figure out the charge at the specific pHs. So if we are at a pH of 1, that means everything is protonated. Well, this is already protonated, this is already protonated. I just need to protonate this guy. Okay, my overall charge is going to be plus 1, neutral, neutral. So that would be plus 1. Now, I'm going to go to a pH of 7. What changes? Well, my alcohol group in my carboxyl end has a pKa of 2.2, so that's now going to be deprotonated. And so when I add up my charges, I have plus 1, 0, and then neutral, so overall 0. Okay, I'm going to keep going. pH of 10, what does that change? That's going to deprotonate my amine group, so this is going to be NH2. That goes away. If I add up my charges, I have neutral, negative 1, and 0. So I get negative 1, and then I'm going to go to a pH of 11, and that's going to deprotonate my alcohol group on my side chain. I'm going to end up with a minus 2 overall charge. So my range here, and notice you're going to get this pattern every time. You're not necessarily going to start at net plus 1 each time, but whatever number you start at, you're going to increase it by 1 every time. Okay, between 1 and 10, but that's really between the lower two. So I need the average of 2.2 plus 9.2 divided by 2, and I'm going to get 5.7. Okay. So I want you to try aspartic acid and histidine, and you can pause the video. I will give you my answers here. We have 2.65 and 7.65. Okay. Now, we're going to do notes 9, and amino acids, they react to make proteins, and so we could have glycine, methionine, tyrosine, and I believe N is arginine, but don't quote me on that because I'm not looking at my sheet. And the bond between the two, which we're not going to do in this video, is called a peptide bond. And when you form a peptide bond, you lose water. So if you've taken a class like AP Bio, you might call these like dehydration reactions. Okay. So I'm going to skip this page. And I just want to draw. I'm going to skip to page, what looks to be 25 in your notes packet, but make sure it looks like this. I want to draw this amino acid protein group at a pH of 7.4. So what I'm going to do is I'm going to recognize that I'm going to have my amine end, or my amino end. I'm going to have my carboxylic acid end. Okay, and if you are an internal amino acid, you're going to lose some of your structure. Not your R group, but your amine group's going to look a little bit different. So what I'm going to do is I'm just going to draw what I know, and then I'm going to go back and I'm going to look at, so I'm going to have my alpha carbon, beta carbon, and then I'm going to have a nitrogen, hydrogen. So notice I lost the carboxyl end there. Okay, and what I like to do is draw an alpha next to the alpha. Okay, here's my amine group. So I'm going to 1 carbon, 2 carbon. First one's the alpha, then there's the beta. And then after my beta, I'm going to get an amine. That is my valine. I'll draw that on there. 1 carbon, 2 carbon. First one's always alpha. And then we're going to draw an amine. My beta carbon always has a CO double bond. I'm going to do it again. 1 carbon, 2 carbon, then an amine. And I have 1, 2, 3, 4 so far. I'm going to write a little bit smaller to get the rest of these on. 1 carbon, 2 carbon, then an amine. And then we have 1 carbon, 2 carbon, and if we're at the end, we don't have an amine, we just have a carboxyl end. Okay? And so notice that the way your alpha carbon points is going to go opposite each time. So if my alpha carbon points up there, it'll point down here, then up, then down, then up, then... And I did this wrong, I goofed. And down. Okay. Now, I haven't even bothered with pKa's at all. So, what I'm going to do is I'm going to look at glycine, and I'm going to have to worry about 2.4 and 9.8. I don't have a carboxyl end in glycine since it's my first one on the left. I just have a 9.8 for my carboxyl group, and so, or excuse me, for my amino group. Yeah, 9.8 for my amino group. Okay, so we're at a pH of 7.4. That is lower than the 9.8, so this will be protonated. Okay, glycine is done. When I go and I look at valine, valine doesn't have any side chains, so I don't have to worry about what it looks like. It's just going to look like what it's supposed to, because my amino group and my carboxyl group don't have pKa's. If you're wondering about this, remember this is a pKa of 38, and so under a biological conditions, you're not going to get to a pH of 38. Okay, phenyl alanine, again, phenyl alanine, I don't know what its pKa is, probably not written, but phenyl alanine is just a benzene group, and I've drawn it wrong, so I gotta erase this. Carbon then. Okay. Glutamine. Glutamine, I believe, let's go back and look here. Glutamine has 9.8 and 2.2, or 9.1 and 2.2, so we don't have to worry about that. So 1, 2, and then on the third carbon is where I have my carboxyl group, and then NH2. Serine looks like this. I'm going to double check the pKa of serine. Again, we don't have a side chain, so we're good. Okay, and then methionine, 2 carbons, an S, and then another methyl, and that's called a thioether. We're going to look at methionine because we do have an N group, and for the carboxyl group, it is a 2.1 pKa, and so if we're at 7.4, that means it's better represented with the H off. Okay, because we're above the pKa. So this is actually my structure at 7.4. Now, we're going to do the same thing. We've got 6 here. I'm going to worry about what these are in just a second, and then I'll let you start on your homework once I finish this. We have, I'm just going to say H3N, carbon, alpha, beta, nitrogen, hydrogen, 1, 2, alpha, beta, nitrogen, hydrogen, alpha, beta, nitrogen, hydrogen, alpha, beta, nitrogen, hydrogen, alpha, beta, nitrogen, hydrogen. So 1, 2, 3, 4, 5. And I need to go down. And I'm going to have to adjust these. Right now I'm just saying plus and minus just because that's what it typically is, but we really don't know until we test the pH. So P, that's going to be a very strange one. That's going to be proline, I believe. And proline, where is proline here? 10.6 is the amino group, so it will definitely be protonated. Now, here's what's weird about proline. Proline makes a 5-membered ring and always has 1 less H. So proline actually looks like that. Nitrogen has 4 bonds, so it'll be positively charged. We have T. T is probably tryptophan, but we'll see here. Threonine. Okay, I was wrong. Threonine doesn't have a side chain, and since it's an internal amino acid, we don't need to worry. And so threonine just is kind of like valine, but okay. K, let's look at what K is. Okay. K is lysine. Okay. Lysine has a pKa of 10.5. Okay, so lysine is 1, 2, 3, 4 carbons in an amine group. 1, 2, 3, 4. 1, 2, 3, 4. And then an amine group. And since it's a pH of 2, that's way lower than the pKa, so it'll be protonated. H is going to be another histidine. And so what happens with histidine is, I'm going to draw what I know it looks like, and histidine's always kind of hard to draw. A I've got to figure out the pKa of this H. So I'm going to scroll back up, and I know that gets annoying, but 6.0. So it'll definitely be protonated because we're at a pKa of 2. Remember, when you're at a lower pH than the pKa, whatever group you're discussing will be protonated. Okay, E. Let's go look at E. E is glutamate, or glutamic acid. That'll be 4.1, so that'll be protonated. Okay. So that looks like 1, 2. Okay, that'll be protonated because we're at a pH of 2. And then our last one is methionine. Okay. And then the thing I need to worry about is this OH. It probably has a pKa for methionine like 2-ish, so we've got to look at it and make sure. Methionine 2.1, and so we're at 2, so it'll be protonated actually. So this will be an OH group. Okay.